For today, we're going to be talking about introduction to toxicology and critical concepts. And we have some objectives that I'll walk through here. And just wanted to remind everyone that these slides are also posted on the Canvas page. So if you would like to follow along on your own slides and take notes, feel free to do so. I'll also be showing the same slide deck that you have available and going through it throughout my talk today. Okay, so the first objective for today's discussion is that we will define toxicology and how it connects with green chemistry and product stewardship. Next, we'll describe adverse health effects, what that means, and how toxicants can cause adverse health effects. We'll explain dose response and talk a little bit about what that means. We'll talk about the concepts of NOEL and LOEL, and don't worry, I'll get into what those acronyms stand for and what they mean in terms of toxicology and evaluating chemical risks. And we'll also discuss ADME, A-D-M-E, that stands for absorption, distribution, metabolism, and excretion of toxicants. And so those are aspects of toxicology that are really critical in understanding how chemicals get into and move around the body. All right, so we're going to jump into the first objective, which is defining toxicology and talking about how it connects with green chemistry and product stewardship, which is why we're all here. So toxicology is actually a pretty broad interdisciplinary field that connects that connects biology, and it connects chemistry and biochemistry, all of these things. And what we're trying to do is really study the effect of toxicants on the body. And a toxicant is anything that can be toxic to the body. That can be chemicals. That's the primary focus of what we'll be talking about mostly throughout this certificate program, as it's focused on green chemistry. But toxicants can also be biological agents or even radiation. And toxicology is known as the science of poisons. We're talking about how these toxicants, so chemicals, radiation, how they can poison people and cause health effects that are not ideal in the body. So you'll notice that there's a picture of an older gentleman to the right of this slide, and that is a person named Paracelsus. He was some call him the father of toxicology, and he had this very poignant quote that has been referenced in toxicology since the 1400s and 1500s. And he very poignantly put that, or really discussed that the dose is what makes the poison. So it's not always about what the chemical is or what the toxicant is, but it's how much of it. And we'll talk a lot more about how that can connect with what we'll be studying throughout this certificate program. And speaking of the certificate program, toxicology is really foundational in when we're talking about green chemistry and chemical stewardship. So what toxicology does is it tells us what health effects a toxicant can cause and at what doses, so that is how much of the chemical or toxicant is needed to cause those health effects. Toxicology allows us to evaluate human and environmental health risks from exposure to chemicals. And human and environmental health risks are one of the foundations of how we determine which chemistries are green, which might be greener, and what their impacts might be on human health and the environment. So I have posted on this slide here a bunch of definitions, and I'm not going to read through them line by line, but feel free to pause this lecture and read through them if you'd like, or go back and reference them as you review these slides on your own. But some key ones that I want to talk about, or that I want to point out, just because they'll be quite relevant throughout the rest of this talk today, are toxicant, and you've already heard me use this term. A toxicant is an agent, so a chemical or radiation that can cause a toxic effect. Sometimes you see the word toxin used when referring to things like this, but toxins are actually a subclass of toxicants that includes only chemicals that are naturally produced by plants, animals, or microbes. So if you've ever had poison ivy, that chemical that comes off of that plant is a toxin. Or maybe you've heard of botulinum toxin. That's something that can cause botulism, but that's a toxic chemical produced by a bacteria that can cause toxic effects as well. So those are toxins. Things that are synthetic chemicals are, or even some, most naturally occurring chemicals that aren't produced by plants or animals, are referred to generally as toxicants. So you'll hear me use that word a lot throughout this lecture and then the other 2 lessons as well. The dose is, this is a word that you've probably heard before. The dose, when we're talking about toxicants, is the amount of the toxicant that enters your body. And what's key about the term dose, and we'll talk more about this later today, what's key about dose is that a chemical has to be inside your body to cause a health effect, which seems intuitive, but but there's a lot of things that we have to consider when we figure out how much that dose is. An exposure route is the way that a chemical or particle or any type of toxicant can enter your body. The primary exposure routes that we talk about in toxicology are inhalation, ingestion, and dermal exposure, that's skin exposure. So you can inhale something. When you breathe it in, you can ingest it by taking it in through your mouth or drinking it through water, and then dermal exposure. Exposure duration and frequency, how often a person or organism is exposed to the chemical. An exposure pathway is the way by which an organism gets to be exposed to the toxicant. And adverse health effects, we'll talk more about this in a slide or 2, but those are really ways by which the toxicant can affect your body and affect your normal physiological functioning. Human health risk assessment is the way that we evaluate the potential for health effects to happen from exposure to toxicants. And much like that, ecological risk assessment is evaluation of the potential for adverse effects on ecological receptors, whether that be plants and animals or the other aspects of the environment as well. All right, objective 2 for today's lesson is describing adverse health effects and how toxicants can cause them. So toxicants and adverse health effects. So an adverse health effect is generally defined as a change in either the morphology, the physiology, growth, development, or lifespan, or many other things that can happen as a result of many things. When we're talking about adverse health effects from exposure to chemicals or toxicants, we're talking about adverse health effects that are caused by those toxicants. Adverse health effects result in impairment of a person or an organism's functional capacity or performance, and reduces the ability to respond to stress or increase susceptibility to other influences in the environment or in their environment. Adverse health effects can be experienced in both humans as well as ecological receptors, such as other species, animals, plants, many environmental systems. There's lots of things that can go wrong from exposure to toxicants. So as we're talking about adverse health effects, what we really want to lay a foundation of is normal human physiology and biochemistry. So what is the baseline and what are we looking at when we talk about, if chemicals are able to cause these adverse health effects, what are they messing up, for lack of a better term? So humans and most other organisms are pretty complex, or many are complex multicellular organisms. We're built of cells that are really the building blocks of human life and other life as well. Cells are arranged into tissues, and tissues are organized into organs, and organs work together in organ systems. And I'll talk a little bit more about how all these things connect. But I really wanted to take it from the starting building block and build it all the way up to how these organ systems work, because then we can talk a little more about how those organ systems can get disrupted from exposure to toxicants. So organ systems, they normally work together to carry out processes that keep us alive, functioning, and healthy. Organ systems make up organisms, and there's lots of complex processes and biological reactions from cellular level to the organism level. So I wanted to walk through, you'll see on the bottom of the slide here, there's a few images that all connect with each other. But it's a good example just to run through, how these individual or how cells, which you'll see all the way to the left, starting with a muscle cell, those cells together, so this example is muscle cells. Many muscle cells together make up muscle tissue. And so that tissue is a functioning component of an organ, which is, in this example, a bladder. So a bladder has, this is a urinary bladder, and the bladder has many different components to it. It's got, some structural, cellular structure, and endothelium and epithelium, but it also has muscle tissue in it. So this muscle tissue is just an example of one of the types of tissues in an organ, which is the bladder. And then the next image that you'll see to the right of that is how that bladder connects with other organs. And don't worry if this, you don't have to focus on the complexity of how these organs work together. But what I wanted to show here was that the bladder is a component of a larger organ system. That is, it connects to the kidneys and other pieces of the urinary system that work to remove waste from the body. And so all of those things together, so kidneys are another organ, and all those organs work together to excrete waste from the body. So these and all of these organ systems, as you know, can work together in a healthy functioning person, and toxicants can have effects, whether it be at the cellular level or all the way up to the organ system level, to disrupt that. We'll talk more about how that happens later, but I just wanted to lay a foundation of, a baseline of how these things work in a normal, healthy manner. So many organ systems, we have many organ systems, and they all work together to keep us alive, healthy, and functioning. So this is just an example. This image just shows a bunch of different organ systems that we might talk about, and we'll talk about a few of these today and some in future lessons. And they all work to perform different functions that our body needs to survive. So you'll see, starting over on the left, there's the respiratory system that's a bunch of organs that work together in order to keep your body oxygenated and expelling CO2. You've got your digestive system, your lymphatic system, endocrine system, which is hormonal systems, your circulatory system, pumping your blood around, nervous system, muscular system, all of these things working together to keep you alive and functioning and healthy. So toxicants can go on to cause disruption in those healthy functioning organ systems. This is another long wordy list, and this time I actually won't read through all of them, but there are many different types. You can classify toxicants or chemicals based on which types of organ systems they can disrupt. So a developmental toxicant is something that can cause disruption in development. There could be an endocrine disruptor, which disrupts that endocrine system. Neurotoxicants are toxicants that can mess with your nervous system. And basically all the way through that image that I showed you of all the different organ systems, there is essentially a chemical class or a toxicant class of chemicals that can disrupt those systems. So when we talk about toxicants and how they can cause these adverse health effects on these organ systems, you know, we might ask ourselves, how do we know which systems they cause health effects to, and how do we know how they're doing it? What are the chemicals doing to cause those effects? And in toxicology, there are these 4 main ways that we test toxicants and evaluate whether or how toxicants can cause adverse health effects and in what systems. They're very, well, yeah, I'll just go through them all here, and then I'll dive much more into each of these as we move through the next slides. So the first step in evaluating, these aren't necessarily steps also, but they're often approached stepwise to really characterize toxicological effects. So the first is structure-activity relationships. And what a structure-activity relationship approach is it's evaluating a chemical's structure, that is its size, its solubility, its stability, volatility, things like that, and using that to determine how a chemical might cause health effects. Another approach within the structure-activity relationship processes includes using those structures, or if you look at a chemical structure, using that structure and comparing it to other chemicals that you know to be toxic to, say, the nervous system. So if you have 2 chemicals that look really similar in their structure and they have similar volatility, and you compare the 2, and this one causes a certain type of health effect, it'd be pretty safe to say, or a good starting place to say, maybe this chemical also causes that because they look so much alike. The next type of test, a toxicological test, is called an in vitro test. And in vitro tests are essentially cell culture experiments. If you've ever seen like a cell culture dish with cells growing in them, this is what this type of test is. And those are used to evaluate a chemical's ability to cause certain types of adverse health effects. We use these to determine potential mechanisms, so that is figuring out how the chemical causes the health effect, and what cells, or how the chemicals are disrupting the cells in those systems. The next type of test is called an in vivo test, or also animal bioassay. An in vivo test, so in vivo means in life, and so those are tests that are done in animals that evaluate a chemical's ability to cause certain types of adverse health effects. These types of animal tests are done using the exposure routes will be those that are relevant to humans when they are evaluating this. So, for example, if there was an investigation to determine whether a chemical caused a certain adverse health effect when a person inhales it, when they do that, the animal bioassay, the evaluation will be an inhalation test, so where animals would inhale the chemical, as opposed to if the question was about something related to whether a chemical was toxic by being ingested, or that is taken in through the mouth, the exposure would be through ingestion. And we'll talk a little bit more about why that's relevant. in these types of in vivo tests. These in vivo tests are often used to determine doses at which health effects may occur. In vivo tests are, so it's a lot, not easier, but it's a functioning biological system, such as a living animal, is a lot more representative of how a chemical will actually affect a person than, say, a dish of cells. When these tests are designed and performed, they are they're not done just out of nowhere. They're very well planned, and there's many layers of ethical considerations taken into account, and they are very well informed from other types of tests. So they'll often do, toxicologists will do structure activity analysis before doing any type of animal bioassay. And they'll also do in vitro testing in cell cultures to make sure that they really have their pencil sharpened and are able to use to test these, do these in vivo tests in a way that are not going to use any animals that don't need to be exposed to chemicals. They will avoid that and do the most ethical testing that they possibly can. Epidemiology is a type of test that is actually more based on observations seen in humans from real-world chemical exposures. And I don't mean that to say that there are toxicologists out there that are exposing people to chemicals intentionally. They're not intentional exposures, but they're largely observations from, I call it in this slide, lessons learned from the past. Sometimes epidemiology studies can come about because there's certain types of exposures, or you see a cluster of a certain type of health effect come up, and you can use epidemiology to determine common chemical exposure among all of the people that developed that health effect. And then you can use an epidemiology analysis, an epidemiology study to determine, okay, yes, that chemical exposure really did cause that health effect. And it shows us that these are really powerful studies because they show us that these health effects really do happen in people. To go back to the in vivo testing that's done in animals, there's always a question, you know, a person is a different type of organism than an animal is. And so while it can be a pretty good representation, there's always a question as to whether, you know, the exact dose that causes a health effect in an animal, will it be the exact same dose that will cause the health effect in a person? And so epidemiology studies help us understand that these health effects really do happen in people. And again, we don't go looking to intentionally expose people to chemicals in order to get that evidence, but it's some things that just happen from lessons learned in the past. And I'll show some examples of each of these types of tests in the next few slides to help give you a better idea of how these things come about and how we study them. So structure-activity relationships tell us about how chemical structures relate to each other, and they tell us a lot about chemical toxicity. So I put on the slide here two very different chemicals, and so I just want to talk a little bit about each of them. I'll start with chemical 1. So chemical 1 is PFOA. And so PFOA, you might have heard this before. It's a surfactant. It's in things that are non-stick. You might have heard of Teflon, things like that. And PFOA is a very large chemical. It's got a really heavy, large structure and is a really heavy chemical. PFOA contains many carbon fluoride bonds that, so if you haven't looked at a chemical structure in a while, I know some of you are chemists, but I'll just walk through quickly that all the branches along that chain are represent carbons, and the little F's stand for fluorine. And so the carbon-fluorine bonds don't break down very easily in nature. It's very hard for those chemicals to break down once they get into the environment. Because it's such a big, heavy chemical, they're not very volatile, so they don't get up into the air, into a place where you can breathe them in very easily, at least in consumer applications and things like that. PFOA actually has a really similar structure to PFAS, which we know is very toxic. So you can look at these two chemicals. I'm gonna turn on my laser pointer here. So we're looking at PFOA and evaluating its toxicity, and we know, let's say we know PFAS is very toxic. So what we'll do is we'll say, oh, these two look really similar. Maybe they cause the same health effects. So if we want to investigate the toxicity of PFOA, we might first look at the literature, the scientific literature, and evaluate what health effects PFAS can cause and use that to inform our assessment. I talked a bit about how these chemicals are not volatile and that they have very large, heavy structures. And so those aspects of the chemical structure can really inform how people might be exposed to the chemical. Something that is really heavy and not volatile is not likely to be inhaled in many of its applications unless it's easily dispersed into air. So that can also help us in our assessment of which exposure routes, if you remember that from the beginning of the lesson today, which exposure routes we should consider. So the second chemical here that I have listed, shown on the right, is toluene. And toluene is a very small chemical. It's very light. It's quite volatile, which means, again, it will go into air very easily. And it has a very similar structure to benzene. So in the same way that we were evaluating PFAS and PFOA, if we wanted to evaluate the toxicity of toluene, we might look at a chemical that it's chemically similarly, structurally similar to. So benzene, we know that benzene is very toxic. It causes many health effects. So if we wanted to evaluate how toluene can cause health effects, we might look to the literature to evaluate benzene. And in a similar way that we evaluated PFAS and PFOA, we would look at the volatility of toluene and benzene, and we would say, oh, okay, these are very volatile. They get into air very easily, so people might be exposed by inhalation as opposed to ingestion or otherwise. So the next test I'm going to dive into is called, is the in vitro test. And this is a very busy slide, but just stay with me and we'll walk through it step by step. This slide is showing what's called the Ames assay, which is a test for mutagenicity. And mutagenicity is the ability of a chemical to cause mutations. So we're going to start over here, and I'm going to use my cursor to point through this, but you can also see that I've numbered each of these steps, essentially, so you can follow along that way as well. So what happens in the Ames assay is we take a test tube, say, of this Salmonella strain of bacteria. And this strain of Salmonella it requires this amino acid called histidine to survive. So it needs histidine. And what we'll do then is we're going to get 2 of these cell culture dishes, and one of them we're going to add a chemical. We're going to add Salmonella to each of these dishes, and to this one up here we're going to add a possible mutagen. So this is the chemical that we're trying to figure out whether or not it can cause mutations. So we add the mutation or the mutagen here, and we add it with salmonella plus the chemical in this plate. In this plate, we're going to add only salmonella. And remember, these salmonella require histidine in order to survive. And then what we'll do is we incubate the cell culture dish, which just means let it rest and grow if possible. And what we'll evaluate at the end of the test here at number 4 is whether any salmonella grew on this cell culture plate. And the way that we evaluate whether this chemical was able to cause mutations is we look at the number of salmonella cultures that were able to grow. So if the chemical is able to cause mutations, the salmonella will mutate and then they'll be able to grow without that histidine. So if you remember, we needed the salmonella needs the histidine to survive, but then this chemical allowed it to mutate so it didn't need histidine to survive. So you have the successful growth of salmonella. In the plate where we only added salmonella with no chemical, so no help in mutating at all, we'll see very little salmonella growing because no chemical was added and thus there were no mutations. And this is just an example of a type of in vitro test that we can use to evaluate whether chemicals can cause certain types of health effects. There's lots of different types of in vitro tests that we can use to test lots of different types of chemical effects. This is just one of them. So the next type of test that I am going to walk through is the in vivo testing, and that's the animal bioassays. And again, I'll walk through this slide starting from the left to the right and talk about how these types of tests are performed. So you'll see on the top left over here is our test group. And there's also a control group. And again, the amount of animals and the number of tests that are done are very tightly controlled by ethics boards, and also the smallest number of animals are used in order to ensure that they can achieve statistical significance in evaluation of the chemical, but that there's no more than the minimum number used. So what we'll do if we want to evaluate a chemical to determine whether or not it can cause a health effect, is we'll have a test group and a control group. And the test group, they will be exposed to whatever chemical is being evaluated. In this case, the chemical is this little yellow dot. And the chemical will be added to, say, their food or their water if the test was looking at ingestion. So that's the taking in through the mouth of a chemical. And the control group will not have the chemical exposure. And the test will take place over a certain period of time that can vary depending on the test. And then if statistically significantly more animals have the health effect in the test group compared to the control group, in this case, the health effect was turning purple. So if more animals have the health effect in the test group compared to the control group, we can conclude that the chemical being evaluated is what caused these animals to turn purple or have that health effect. So epidemiology studies, again, those are the evaluations of health effects in populations. If you remember, I referred to this almost as like a lessons learned from the past type of evaluation. There's lots of different types of epidemiology studies. It's not even always necessarily looking in the past. It can be looking forward also. But I wanted to just show an example of a type of epidemiology study that has been used to evaluate whether certain types of chemicals or toxicants can cause health effects in people. So in this example, I have an example here of, at the top, we have, these are a bunch of miners working in an asbestos mine. And at the bottom here, we've got a bunch of miners that are working in a lead mine. And so what would happen in an epidemiology study, I'm gonna move my little face over here. What would happen in an epidemiology study is, say we were observing these populations of people, and we found that of all of these people that worked in the asbestos mine, a whole bunch of them developed a certain type of disease. In this case, the disease is turning green. So in the asbestos group, we see that a large number of these miners developed this health effect. In the lead mining group, you'll see that only one of the same size population developed the disease. And what an epidemiology study would then do is use statistics to determine if statistically significantly more workers have had the health effect compared to the other group. And if they do, then we can conclude that the difference in exposure between the two groups is what caused the health effect. You'll notice that these two groups that we're comparing are really similar. They're both miners. They're both groups of miners. They're both working similar types of hours. They're both working inside these probably not very well ventilated areas. The only difference between these two groups is that this population is working with asbestos, and this population is working with lead. And so that really helps us whittle down, because people are really complex and dynamic people. We live really complex lives, unlike in the in vivo studies where mice are very well controlled and their diets are very well controlled. Studies in populations can be pretty hard to control. So as close as the control group, or the two groups that we compare, we try to make them as similar as possible. And again, this is just one type of epidemiology study. You will notice that this person in the group that did not have the exposure that was the difference in the exposure, this person also developed the health effect, which would seem kind of strange. You'd say, okay, well, if asbestos is what's causing these folks to turn green, why is this guy turning green? And I alluded to before that, people live really complex lives, and it's really hard to control everything that people, we don't really try to control everything that people are doing when we're, evaluating whether health effects can happen from exposure to chemicals. But it's possible that maybe this person lives with someone that works in this mine. So say this person lives with this person, and this guy is coming home every day with asbestos on his clothes and shaking it out, and getting it all over the house. And so this person goes on to develop the disease as well. And that just really speaks to the power of statistics. And you'll also notice that these groups, these populations that we're studying, are much larger than the groups of animals that are involved in the animal testing, because we really need to have a large number of people in order to account for the differences in or just complexities in people's lives. And again, I just want to clarify that no one is intentionally exposing anyone to these chemicals, and that's not that's not the way that these studies work. They're just observations generally in people that are living their lives normally and as they were. And sometimes health effects come up, and we use statistics to figure out what caused them so that we can have interventions to reduce those health effects or reduce those exposures so that people don't have the health effects anymore. So in this study, let's pretend this was way back in the day, before anyone knew that asbestos could cause mesothelioma. Once they figured this out and they said, hey, all of these people are getting mesothelioma that work in the asbestos mine. All these people that work in the lead mine don't get that. Maybe they could make some type of intervention that says you have to wear respiratory protection when you go into an asbestos mine, or just don't mine asbestos anymore. So there's lots of different things and public health interventions that can happen once we figure these things out. Okay, the next objective for this lesson that we'll talk through is dose response, NOEL and LOEL concepts. So dose response is, it's called the dose response relationship. And basically what the dose response relationship is doing is it's describing the relationship between the level of exposure to a chemical or a substance and the likelihood of an adverse health effect associated with that exposure. Generally speaking, and this isn't always true, as the dose increases, so the amount of chemical that you get into your body, as that increases, the severity or the frequency of occurrence of the health effect will also increase. If you remember from our friend Paracelsus, the dose makes the poison. So more chemical exposure means larger health effect. You'll see on the bottom of the slide here, I have a dose response curve. It's called a dose response curve when we're looking at the dose and the response being the health effect. And we're looking at the relationship as the dose increases, does the health effect or the response increase? I'm going to talk through how we would go about getting this type of figure from a toxicity test. Okay, so if you remember back to the slide that we talked through, the in vivo study, we were trying to figure out if a certain chemical caused a health effect when, say, these are mice, when mice were exposed to it. And we want to know that because let's pretend that this is a chemical that we want to use as a, a water treatment agent. So we want to make sure that it's not going to be harmful to people, and it's something that we want to evaluate for toxicity beforehand. So what toxicologists would do to evaluate that is they would develop this type of study. Again, this type of study would be very well informed by previous studies and evaluations of chemicals that were similar to it in those structure-activity relationships and cell culture assays to make sure that we were really testing the right thing and not doing frivolous research for no reason. And they would come up with a test plan where we would compare different doses of chemicals to each other. So we would come up with these different dose groups. So the first dose group here on the left is 0 milligrams per kilogram per day of chemical. So essentially what that means is this group is not being exposed to any chemical. The next dose group is going to be exposed to 10 milligrams of the chemical, and that's per kilogram per day of the chemical. So just a little bit in this water bottle is how they're being exposed. The next dose group is going to be exposed to 100 milligrams per kilogram per day, so a little bit more. And then the last dose group is going to be exposed to 250 milligrams per kilogram per day. So you'll notice as we go up, we're increasing the dose that each group is being exposed to. And what we would see in a very simplified toxicology study is the control group. So those that didn't have the chemical exposure would have no health effects. Those that were exposed to just a little bit, only 10 milligrams per kilogram per day, of the chemical would have, not that many of the mice would have the health effect. In the next dose group, we'd have a little bit more, and the next dose group, we would see more. And you might wonder why, if this guy doesn't have any health effect from exposure to even this much of the chemical, why did this guy have it? And so the reason that happens is because there's, even though when these tests are done, these mice are very similar to each other, they're all eating the same food, they all live in the same space, there's still genetic variability between animals. And so every mouse is going to be a little bit different from the other one, and maybe this one was just a little more sensitive to the chemical than the other ones. And maybe this guy was real hardy and just, could withstand exposure to that amount of chemical without having the health effect of turning purple. Generally you see these dose response curves of increasing response or health effect from exposure to the chemical. And so this is the slide, I showed this previously, but you'll see now, the study that we just talked through, we could then take those findings where we've got 0 out of 4, 1 out of 4, 2 out of 4, and 3 out of 4 mice having the health effect, and we could plot that on a graph, and we could draw that dose response curve. So you'll see here, we've got the 0 with the 0, a dose of 0 with 0 health effect. The dose of 10 milligrams per kilogram per day has 1 out of 4. The dose of 100 has a 50%, so 2 out of 4 had the health effect. And at the dose of 250 milligrams per kilogram per day, we had 3 out of 4 of the animals had the health effect. And so this together can make a very nice oversimplified dose response curve. But what does that mean? How do we take that and these findings and connect it to how a person could have health effects from these chemicals? So what we do next with this type of information is we will try to determine, using statistics, what the NOEL is, or the no observed adverse effect level is. And the NOEL, the definition of a NOEL, is the dose in toxicology study at which no adverse health effects are observed, or no statistically significant adverse health effects are observed. So in this study, you'll see that in the results I indicated here, there's this little star asterisk to indicate that there's a statistically significant response compared to the 0 dose, or that control group. So in this example, let's just say that dose group of 10 milligrams per kilogram per day, that was not statistically significantly different from the control. I hope no one's checking my math, because this is just an oversimplified version just to show you the definition and the application. But at the 100 milligrams per kilogram per day, this was determined to be statistically significantly different from the control, and as is the 250 milligrams per kilogram per day. So when we're looking at the NOEL, we want to look at the dose at which no adverse health effects are observed, or no statistically significant adverse health effects are observed. So how do we determine that NOEL? What we do is we take a look at the dose response curve, and we find the dose where there's no statistically significantly different responses that compared to the control. In this study, it was that the dose that was the dose where there was not statistically significant difference from the control was 10 milligrams per kilogram per day. And so what we do, so we find this point on the graph, and we would follow that down and determine that, okay, 10 milligrams per kilogram per day is the dose that had no statistically significantly adverse effects observed. So in this case, the NOEL in this study would be 10 milligrams per kilogram per day. Similar to the NOEL is this concept called the LOEL, and the LOEL stands for the lowest observed adverse effect level. And the LOEL, the lowest observed adverse effect level, is defined as the lowest statistically significant dose in that toxicology study where we do observe adverse health effects. So what we do then, in this case, just like when we're evaluating the NOEL, is we find the point on the graph where we see the lowest dose that caused the health effect to be observed, statistically significant health effect. So what we do is we identify that point so that this is the lowest dose with the statistically significant effect here, given that asterisk. And we follow that down to the dose axis, and we find that the LOEL is 100 milligrams per kilogram per day. So you might be wondering, how are these NOELs and LOELs used in toxicology? Why do we bother to evaluate them or figure out what they are? And so the NOELs and LOELs, those tell us the levels at which health effects can happen. NOELs and LOELs from these types of toxicology studies can be extrapolated to determine where health effects might occur in humans, which is really, I mean, when in human health risk assessment, especially, this is what, the ultimate question is. We do these toxicology studies to make sure that health effects don't happen in humans. So these human levels are the foundations of what's called a toxicity threshold. We'll talk a lot more about that in later lessons in this series, but I just wanted to provide the context that these toxicity thresholds are levels at which health effects are not expected to occur. So toxicity thresholds are established by various different agencies. You might have heard of the US EPA, Environmental Protection Agency, the OSHA, that's Occupational Safety and Health Administration. Those groups establish these toxicity thresholds that tell us where we can safely be exposed to chemicals without having health effects. Some of those examples of those toxicity thresholds are known as reference doses or occupational exposure limits. I'm not going to talk very much about those now. We'll talk about them in lesson 5. But I just wanted to provide that context of where these, where the toxicology studies are taken, the next steps after we get these results from them and how they can inform health assessment. So the last concept that I'm going to talk through today is the ADME or ADME, and that stands for absorption, distribution, metabolism, and excretion of toxicants. So what ADME is really talking about is what happens to a chemical, or what happens when you're exposed to a chemical. I'm going to walk through these step by step, but just briefly, I'll go through this little figure right here. When you are exposed to a chemical, you will absorb the chemical into your body. The chemical will be distributed around into your body, into different tissues and organs. And the chemical will be metabolized or broken down, and then it'll be excreted or released from your body. So absorption is really talking about how a chemical gets into your body. So there are three primary exposure routes that we think about when we are evaluating toxicology concerns. The first being ingestion. And ingestion, again, is absorption or is the taking in of the chemical through essentially just through your mouth. That can be through drinking water, as shown in this example. It can also be ingested through food, or even sometimes it can be through getting chemicals on your hands and touching your face and your mouth. You can get chemicals into your mouth that way. And so when you ingest a chemical, absorption can happen in your stomach or in your GI tract, in your intestines. And that is the primary way where where chemicals that are ingested get absorbed into your body. When you inhale a chemical, again, inhalation is breathing chemical in, you absorb the chemical through your lungs. And from your lungs, it gets into your bloodstream, and then it can be distributed around your body from there. And dermal exposure, or skin exposure, is absorption through the skin. And some chemicals can be absorbed through the skin quite easily. Some cannot be absorbed through the skin very easily. But when they're absorbed into the skin, your skin's connected to your bloodstream. So your skin or the chemical that gets in through your skin can then get distributed and go on to, be distributed throughout your body. An interesting concept, which is kind of a distinction between exposure and dose, is that not all of a chemical that that is in an exposure will get absorbed. So the chemical that gets into your body is what can go on to cause the health effect. An example that might help conceptualize this is, imagine if you had a glass of water and you had a chemical in it, and you drank that water, and some of the chemical will get absorbed through in your stomach into your bloodstream. And when the chemical that got in would be able to go through and get all into your other organs and could go on to cause a health effect. But some of that chemical that you ingested doesn't ever get absorbed into your body. It just goes right through your stomach and goes through your intestines, and you excrete it before you ever absorb it. So distribution, that's the D in ADME, that talks about where the chemical goes after it's absorbed into the body. So different chemicals distribute to different types of tissues throughout the body. That distribution, where the chemical likes to go, is actually based on the chemical properties itself. So some chemicals that are lipid soluble, or they prefer to be in tissues that have fatty structures, those chemicals will end up in your fat or in organs that have a lot of fat in them. Some chemicals stay in the bloodstream a lot longer. Those chemicals are usually water-soluble chemicals, and they don't distribute into fatty tissues as much, but they can find their way into other tissues as well, just depending on their chemical structures or physical chemical properties. And yeah, those are really key in figuring out where chemicals go when they get distributed into the body. Metabolism describes how chemicals are broken down inside of the body. So chemicals or cells contain these chemicals that are called metabolic enzymes. And what those metabolic enzymes do is they break down chemicals. And the liver contains a large number of these metabolic enzymes. So that's why you'll see in the figure here the lots of chemical in the liver. And what the metabolic enzymes are doing is they're really reacting with chemicals to try to make them easier for your body to excrete them or to get them out. Your liver acts like a filter, and because it has so many of those metabolic enzymes in them, it will really be really efficient at removing a lot of those chemicals from your body. And when your blood is flowing around through your body, through your bloodstream, your blood passes through your liver all the time. And that's great because your liver is constantly filtering out these chemicals or chemicals that might have gotten in to your body and metabolizing them and helping them get excreted faster. And excretion is really talking about how the chemicals leave the body. As I was just talking about, metabolized chemicals are made to be, or chemicals are metabolized. And what these metabolic enzymes are doing is making chemicals more water soluble. And making chemicals more water soluble is really good for excretion because that helps them get into the urine. And the urine is a really efficient way for your body to get rid of chemicals. Fat-soluble chemicals, you can imagine they're a bit harder to get to be water soluble because if you remember, like oil and vinegar, they don't mix very well. And so fat-soluble chemicals are a bit harder to be made water soluble, but there are other pathways by which and mechanisms by which fat-soluble chemicals are excreted, primarily through the bile, and actually are excreted. Bile comes from your gallbladder, and that will actually excrete the chemicals into feces. Some chemicals, when they get distributed into your body, they'll stay in there for a long time before they're excreted. That's really dependent on the chemical itself. And whether it's something that is really efficient at being stored in a certain type of tissue. Some fat-soluble chemicals will stay in fat for a really long time, and it takes a long time for them to slowly get moved out. So it depends on the chemical, and we'll talk a bit more about that throughout this course, actually. And how those chemicals can have persistent effects on the body. So just to go through again, these are the objectives that we talked through today for lesson 3. We talked about the definition of toxicology and how it connects with green chemistry and product stewardship. We talked about what an adverse health effect is and how toxicants can cause them. We talked a lot about different types of toxicology studies and how those can inform potential effects on humans that can also be applied to potential effects on animals or other species. We talked about the dose response concept and how, generally speaking, the greater the dose, the greater the response is, and greater the health effect is. We talked about the NOEL and the LOEL. And we discussed ADME, that was the absorption, distribution, metabolism, and excretion of toxicants.